Okay, welcome back, ladies and gentlemen. Hope you had a good weekend. We're going to continue with DNA replication. So last week we started with initiation. We kind of stopped there. So we went through kind of initiating DNA replication, but we didn't actually replicate it. Today we'll kind of finish the replication. Okay, so we're going to go through the second and third stage of elongation and termination. So just a summary, the DNA replication happens in three phases. They're called the initiation phase, the elongation phase, and the termination phase. The termination phase is really nothing, okay? There's nothing really, it's like one slide, it's nothing. It's really the initiation phase and the elongation phase. So let's recap the initiation phase from last week. I don't know why this picture looks so crappy, but I'm running into a whole bunch of computer problems. So anyways, the big things that you need to know here is what helicase does. So helicase is there opening up, cutting those hydrogen bonds between the bases. Okay, and then we got topoisomerase, which is basically relieving the tension and making sure the DNA doesn't rip apart too early. And then the last thing we had was single-stranded binding proteins, and that's just making sure that the DNA doesn't reform before it's been replicated. Okay, so now we basically, if you look at here, we have this strand right here is ready to be copied, and this strand right here is ready to be copied. They're opened up, and now we're going to start getting some new things coming in to replicate it. So remembering that the DNA is open at this point, just like it is here. And now we're going to go, okay, so we're going to be going through the initiation next stage, which is called elongation. All right, now, during this phase, new nucleotides, like bases, sugars, and phosphates, are added to each of the parental or the original strands. Okay, so the new nucleotides required to form the brand new DNA came from any kind of cells that maybe you destroyed in your body, like if your liver cells die, you'll actually take your old DNA and recycle it, but also mostly from the food that we eat. Okay, we eat a whole bunch of food, we take a plant's, we take a banana's DNA, rip apart all their bases, and then we have them stockpiled, and now we're starting to use those bases to replicate our own DNA. Okay, now nucleotides need to be added only in the 5' to 3' direction. This is not going to be a huge factor. If we were in class, we talked about it a lot more, but just knowing that you can only kind of add nucleotides in one direction. Okay, and then we'll kind of explain why that causes a problem coming up. Okay, because the fact that nucleotides can be only added in one direction creates one strand that's really easy to duplicate and the other strand that's a little trickier. Okay, so this is a good picture. So this is the idea that we have our good friend helicase here, okay, and it's just opening up the DNA. Okay, so it's opening up the DNA and it is going this way. Okay, so it's heading to the left. Okay, so the next part of DNA that's going to open up is this. Now, both the top strand, so both this strand right here needs to get replicated, and the strand needs to get replicated. Both of them are going to get replicated. Okay? And you can see these are parts of the new strands that are forming. These are, brand new DNA. These bases came from the foods that you eat, and they just line them up there. Okay? Now, let me go back here. Since helicase is going in this direction, right? One of the strands, this bottom strand, which we call the leading strand, gets replicated really easily. Basically, what happens is helicase keeps moving to the left, and a new base is just going to keep adding and keep adding and keep adding, and it's just going to follow. Basically, DNA polymerase is going to follow helicase, and as helicase opens up more DNA, it's just going to keep replicating. That's the easy strand to replicate. Okay? The problem is this top area, which we call the lagging strand. DNA polymerase can't go with helicase. It can't go to the left. It actually has to go to the right. So the only way that you can replicate it is basically kind of replicate it, then stop. Come back here, replicate it, then stop. Come back here, replicate it, and then stop. You actually work going away from DNA helicase, and that makes it a little bit more difficult. Right, now, I realize this is confusing, so if you're a little bit lost, don't worry about it. Let's go through it a little picture right here. So let me erase everything that I did here. Okay, so the first thing that's going to happen is we got this DNA open, and that was the initiation. Now, elongation starts with basically laying a primer. So the first thing that's going to happen is we are going to lay a primer. So right here, I'm just going to color this in, basically a primer gets laid. Okay? And basically this DNA polymerase right here, this molecule, found that primer and then located on the primer, and then started working its way to the left, laying bases all the way along, and you can see that's the next base right there that it's laying. It's just going to keep laying bases, and as helicase opens up, it's just going to keep laying and more. So you had to put one primer down originally, and then that's all you needed. Then you just needed DNA polymerase to start adding bases to replicate the DNA. Slower. So the first thing we're going to talk about is the easy one, the leading strand. So these are the steps. Okay, you got your DNA. Go back. So that's step one. A primer is attached to the unwound DNA. Okay. Now, an enzyme called DNA polymerase then locates the primer and begins laying bases in the direction towards helicase. Okay. So DNA polymerase can only lay bases in one direction. So now we go back. So very simple process. Okay? DNA polymerase follows helicase, adding bases right behind it. That's how we replicate the leading strand. A primer is attached to the unwound DNA. An enzyme called DNA polymerase. It's open. Okay. Okay, so this is a good picture of it. This is the leading strand. You can see how nice it's replicated. It's just one solid piece, and helicase is going to keep opening that up, and it's just going to keep replicating. Okay. Now, look at the bottom. This is the lagging strand. This is a mess, and this is why it's a little bit trickier. Okay. The first thing you have to do is you have to lay down a primer. Okay. The difference, though, okay, we're going to have to lay down several primers when we do this. We're going to have to lay down a primer here, and then here, and then here, and then here. Because what happens is we lay down the primers, DNA polymerase finds it, and then starts working back this way. The DNA polymerase is going to find the primer here, and then lay down some more bases. It's going to detach to find the primer here, lay down more bases. Here's the primer. DNA polymerase is going to come in here, and then start laying down bases here. Okay? So what happens is basically you have to make a whole bunch of tiny little segments of DNA on the lagging strand. You basically have to create a primer. DNA polymerase will lay the bases away from helicase, detach, go back, and you basically kind of leapfrog all the way down. So let's go through the steps. What I mean by that is it's eventually going to come to an end. So DNA polymerase keeps adding bases, and then comes to an end. Then it detaches. It's going to come back to this primer, and then it's going to start laying bases until it runs into this primer. Then it will detach, and this is the next spot for DNA polymerase to start adding. And it's going to start adding bases here, 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 and here until it runs into this spot. It's going to have to detach and then land again here. What you'll end up getting is a whole bunch of these little segments. And let me just erase this for a second. So you're going to end up getting a whole bunch of these little segments, which we call Okazaki segments. And all they are basically little pieces of DNA that have all been kind of broken apart because DNA polymerase came and then stopped, and DNA came and then stopped. Okay, it's not like the leading strand, which is all one piece. It's actually a whole bunch of different fragments. So the last thing we have to do is we have to glue them all together. And when we glue them all together, that's what DNA ligase does. Okay, that's the purpose of ligase down here. It's going to go in and basically just glue all the little pieces together. So let's go through steps for the lagging strand. Well, it starts the exact same way. Okay? So an enzyme called DNA polymerase locates the primer, then begins laying down bases. DNA polymerase comes to an end. So then a new primer is added. The enzyme called the polymerase, again, this is the process repeating over and over again. So, and that's the lagging strand. So this is just kind of a big picture of everything happening here. Okay? Now, that's the end of the elongation phase. Now, the last phase is the termination phase. Excuse me. There's not much to say about the termination phase. Basically, as soon as the DNA has been replicated, it winds back into its double helical structure and replication machine, which is all the different enzymes, helicase, ligase, polymerase, and so on, all basically just breaks apart. Okay? And that's the process of DNA replication from beginning to end.